So, as I promised, today we're going to be looking at pedigrees. So, let's start. So, we all know that humans, we could have two types of inheritance that we learn, autosomes. Autosomes, which is just normal inheritance, the monohybrid, dihybrid crosses. We could have two sex chromosomes, that's a X, Y, and, So, there are two types of inheritances. Essentially, autosomal is just the regular one, which we will usually, when we refer to it, we will refer to autosomal recessive or autosomal dominant. However, there's also the sex chromosomes, and when they do the inheritance, we usually refer to them as X-linked or Y-linked. However, Y-linked is a lot less common, so you don't have to worry too much about that. So we know that each gamete produced, well, to say each humans or produced, we will have 22 autosomes and we will have one sex chromosomes. Sorry, each gamete, only a sperm or an egg, it will have 22 autosomes and one sex chromosomes, either X or Y. The sperm will have likely Y, or then the egg is likely gonna have an X. So the zygote, when you have this zygote, you're supposed to have 44 autosomes and two sex chromosomes for a total of 46. However, we do have learned about the diseases and the disorders, which means that you could potentially have a 47th chromosome, and you will be a little slower than regular, but however that doesn't matter. We're looking at pedigrees today. So we talked about autosomal inheritance. So it's basically genes, alleles located on an autosome. So gametes get one of the two autosomes, and inheritance of traits is determined by the genes on an autosome. So essentially we're just looking at, hey, regular monohybrid and dihybrid crosses, it's autosomal. Autosomal recessive, autosomal dominant, it's just regular, the regular dominant and the regular recessive. Not too much. I don't want everybody complicating that. It's not too hard. So the pedigree is a flowchart that uses symbols to show the inheritance pattern of certain traits in a family over many generations, like a family tree of genetics. However, well, the reason why we do this is because we want to know what's the chance of your baby potentially having disease. Because obviously it's not ethical to go ahead and make you reproduce with another woman or a man just to tell the disease. I mean, I don't think it's ethical, but some people might think it's ethical, but because most people would agree upon that they are not ethical to just go ahead and mate and reproduce with a male to see which disease you could have. So essentially we look at what happened before you. So we have this inheritance pattern. So this page is really important. I want you to be able to, well, at least know about this and tell me, because you're going to be looking at a ton of pedigrees in the future, and I want you to know that what the heck is happening in here. So right here, the square is a male and the circle is a female. When you have a line that's linking them together, they're called mating. However, unfortunately, so they could potentially be divorced, but they made it at one point. So let's say if this guy, he divorced with her and made it with another one, you would just cross this line. Let me get a laser. So you would go ahead and cross this out and make him mate with another person and add a bunch of things here. That doesn't matter. So identical twins, they're this. Non-identical twins, they're like this. So remember what I talked about the fertilizing egg thing. If you had produced two eggs, you will become non-identical twins. If the twin came from one egg, you'll be identical twins. And then affected individuals are colored in, while the deceased one is crossed out. So sex unknown. You're like, hey, is that for political correctness? No, people, we don't do this in biology. I know that a lot of people be like, hey, I identify as a Walmart bag, or I identify as a Costco shopping cart. I identify as Costco pizza. No. All right, sex unknown only means that they're trying to have a baby. However, when the baby is still in the mother's stomach, or uterus, we don't know the age. We don't know the sex of that baby. We can't tell yet. And I don't think we're allowed to tell people, because apparently back in the 1980s, people valued a male over a female, which cost a lot of parents when they hear, oh, it's a female, they kill them. So now we're not able to tell people, but whatever. So that's how we get a sex unknown. So these parents, if this person decided to get a mate and they wanted to have a baby, and now she's pregnant, now there will technically be a this in the placeholder, because we don't know their sex, but however we want to predict what's the chance of them getting this certain trait. So this is really important, and you're asked to actually label all of them. So this is the generation 1, generation 2, generation 3, generation 4. So I want you to just go ahead and label it 1, 2, 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 3. That's really simple. So we're going to talk about autosomal inheritance. When using a pedigree to study a certain disorder, you can determine if the pattern is autosomal dominant or autosomal recessive. So if both parents are affected, but the child is not, this likely hints that the disease is dominant. Now you may ask, hey, why? Well, both parents could have been a heterozygotes, so somehow they hit a 25% chance and passed the recessive gene on, so their children is unaffected. So you can see autosomal dominant. However, and we have a pedigree here: affected parents, non-affected children. Huntington disease, I gave that autosomal dominant, so you can see. So the pedigree shows the inheritance pattern for an autosomal dominant disorder. Notice that an affected child must have at least one affected parent. Yep. So essentially, when you're looking at autosomal dominance, when you have a child that is affected, then definitely at least one of your parents is affected. If you are looking at an autosomal dominant and none of your parents are affected, you gotta ask your teacher, hey, what the hell is wrong with this? So now we look at recessive. 2 unaffected children have a child with the disorder. So it means that both parents were heterozygous, and so the gene was hidden. And obviously this hit the 25% chance again, and they passed it on, which is shown right here. Now, this pedigree right here shows the inheritance pattern for recessive disorder. Note that the recessive disease tend to skip generations. So maybe a couple generations, maybe your grandpa had a certain disease, and well, okay, let's say somebody's great grandparents had a disease, and then the grandparents didn't get it, and then your dad or their dad didn't get it, and now we're like, oh my god, I'm so lucky they didn't pass it on. And suddenly they have the disease. It's because it's an autosomal recessive. Now we're looking at a pedigree. So what is the phenotype and genotype? Well, we can't exactly tell right away, obviously, but obviously we're able to make some inferences. So let's see. First, let's determine if this is a dominant or recessive trait. So usually I will calculate how many individuals affected, 1, 2, 3, 4, and how many in total, which is 16. 4 divided by 16, that's 25%. So only 25% is affected. I would really say that it's probably a recessive trait, because if the majority is affected, that could potentially be dominant, but we're looking at a minority here. So we can see that it also skipped generations here, so it's likely going to be recessive. So this parent is likely going to be, let's see. This is probably a full recessive, small a. This parent could be a heterozygote, or she could have been a homozygote dominant. However, on the left, these two, they're both heterozygotes. So we don't know about these, but we know that she could potentially be carrier. So they have it. So it means both of them are a carrier. So if their child has it, but they don't express it, it means that both of them have it. And both of these are carriers. So this is big A, small a, big A, small a. And we don't know about them because they don't mate. They didn't have mate yet, and they didn't have a children. However, this one, we know it's full homozygous recessive, and they have the disease. So these genes, they actually dictate what you're good at. Essentially, how tall you jump, how high you will grow, how long your arms, how fast you run, and all that, it's actually all in your genes. So let's look at this. What is this? Is this the same thing? No, it's not. So let's see. So we have 4 individuals out of 14 that's affected. I can't do that calculation right off in my head. It's around 28%, something like that. So 28.5%, let's say. 28.6. Yeah, 28.57, something. Yeah, 28.6. We'll do that. So I'm actually a math teacher. Okay, no. So, well, we can see it right here. Fast twitch muscles, dominant over slow twitch muscles. So it means that most of them have fast twitch muscles, while some of them have slow twitch muscles. So this is a full homozygous recessive. Homozygous recessive, homozygous recessive. What does that mean? This means both parents are the carrier. So this is big A, small a, big A, small a. And these parents are both carriers. And hold up, since he was a carrier, I'm going to say that this parent is actually a homozygous dominant. Why? So you can see 4 parents. Sorry, these parents made it. So he was fully recessive. They had all kids that don't express the trait, meaning that they at least have one dominant allele. So how big of a chance is that? You know, how big of a chance is that? So I would likely going to say that she is going to be a full dominant because, well, if she is a heterozygote, then the chance of getting a full homozygous recessive, it's going to be 50% chance, which doesn't really show here. So again, pedigree is really mathematical based. If you're really good at math, you would excel in monohybrids and all of this crosses. So yeah, that is the end of the first class. I just wanted to get everybody to introduce to pedigrees, and we will look and analyze and see the probabilities of pedigrees in the next class.